Okay, so in the last uh, video I showed you the direct stain with crystal violet, but we also use the indirect stain or the negative stain for staining bacteria. So here I'm going to do what? I will take the slide first, and then I'm going to write that it is from the tooth area, and this is a negative stain. So I'm going to use Congo red for the negative stain. So first I have to do, I have to cut this long neck of the Congo stain tube. Later I have to wash this scissor. And so what I'll do, I will take the droplet or a small drop at one end of, see, it is this much small, the drop will be at almost one end of the slide, not at the center. Okay, and since I'll be using cotton swab, so I'm taking a little bit more because the swab may take some of the stain with it. But when we do in the lab, we don't use actually cotton swab. In that case, we take only a little small drop of the stain. So now I'll take another sterile swab and we'll take the swab from the tooth area. Let me do that. Okay, now I will try to mix over there. See, it has taken everything, it has soaked up. So let me put more of the Congo stain over here. Okay, now let me try to get rid of the everything from the cotton swab. Yes, I'm trying to get rid of everything so that, okay, now I will take another clean slide and will hold the slide at that end of the slide where I put my droplet of Congo red, and I put the swab of my tooth. Now I will just go a little bit back and then I will come forward. So this is how you have to make the thin bacterial smear. Okay, I'm getting rid of the excess one. Now the whole stain is having the bacterial smear. I will get rid of this in the alcohol, but since I'm showing you the demo, I would prefer to show you one more time what I did, so that might help you. So my Congo red was over here with the cotton swab, and then I took another clean slide and with an angle of 45 to 30 I keep the slide like this. Did you see that? I keep the slide like this. Then with the Congo red and also material, I go back a little bit and then I come forward like this. This is how you have to make the smear. Perhaps many of you have seen that this type of blood smear is made in the diagnostic lab. Okay, so this one will go for the disinfection. And this one is ready for a microscope. Let it be dry a little bit. So this is how you have to make another slide of indirect stain. So in indirect stain, you do the staining of the background. You cannot stain the bacterial cells. The background will be stained and the cells will be appeared as casting, like ghost cells, round shape, then rod shape, spiral shape, or the arrangement, we can say whether the two round cells are together, whether they are diplococcus, whether the two rod cells are together, whether they are in a chain, whether they are in a bunch of grapes, like that we can see various types of arrangements. So the purpose of indirect stain is to study the morphology of the bacterial cells. Okay, so it is almost dried out and you can see it is almost ready. Let me show you in the background how it looks like. It looks like this, but it is good to see them. The making of thin smear is very important in microbiology, because if you don't do the thin stain, then what happens? Then you cannot see the shape or the arrangement of the cells. Okay, so we are done for this exercise 1. Thank you.